In the vast and complex world of cancer research, scientists are pretty much always on the lookout for clues. But what if one of the biggest clues of all was hiding in plain sight? Today, we're opening a case file on a scientific investigation that asks that exact question. We're on the hunt for a single genetic suspect, one that might be secretly pulling the strings behind dozens of the world's toughest cancers. All right, so every good investigation starts by surveying the scene, right? And to really get why finding just one culprit would be such a massive breakthrough, we first have to wrap our heads around the sheer scale and the complexity of cancer itself. 20 million. I mean, just think about that number. That's how many new cancer cases popped up in just one recent year. But here's the thing. The real challenge isn't just that huge number. It's that cancer isn't one disease. No, it's hundreds of them, and each one has its own unique genetic fingerprint. So you can imagine, it's like trying to find one single master key that can open hundreds of totally different locks. And that incredible challenge is where our scientific detective story really kicks off. The hunt for a common thread, a single suspect involved in a whole bunch of different crimes. So after digging through just mountains of data, these investigators, they started to narrow things down, and they focused their attention on one prime suspect, a gene called CCT5. Okay, so what is CCT5? Well, in a normal, healthy cell, CCT5 has a pretty respectable day job. It's part of this tiny molecular machine that helps fold proteins into just the right shapes. And trust me, that process is absolutely crucial for keeping our cells running smoothly. But researchers had a hunch. They suspected CCT5 was leading a kind of double life. See, some early clues suggested it might be moonlighting, helping tumors grow and spread. So they launched this massive investigation, what's called a pan-cancer analysis, to see if the suspect's fingerprints were all over these different crime scenes. So, what did they dig up? Well, the case against CCT5 is pretty solid, and it's built on three really key pieces of evidence. First up, the suspect was found at the scene of the crime. Pretty much across the board, in almost all 33 tumor types they looked at, the CCT5 gene was overexpressed. What does that mean? It means its volume knob was turned way, way up. It was working in total overdrive compared to healthy tissue. This wasn't a coincidence, it was a clear pattern. And this hyperactivity, well, it had some pretty devastating consequences. Evidence number two, there was a direct link between how active CCT5 was and how well patients did. The higher the gene's expression, the worse the patient's chances of survival were. I mean, this wasn't just some random connection, this was a real threat. So that led the investigators to the big question. What's the motive? Why was this happening? Well, it turns out our cells have this built-in safety switch for genes, a process called methylation. You can think of it like a dimmer switch on a light. And what they found was that in cancer cells, the dimmer switch for CCT5 was basically broken. It was stuck on full blast, just letting the gene run completely wild. But wait, the story doesn't just end inside the tumor cell. Oh no, the investigation also uncovered something else. How CCT5 might be manipulating the body's own police force, our immune system. So it looks like CCT5 isn't just an inside man working for the tumor. It's also influencing the entire battlefield. Its activity seems to mess with how our own immune system is supposed to recognize and attack cancer cells. It's basically helping the tumor hide in plain sight from our natural defenses. And they found some pretty specific proof for this. For example, in kidney and stomach cancer, high CCT5 levels were linked to two really key markers for immunotherapy. The first one, TMB, is kind of like a count of the tumor's red flags. The more flags, the easier it is for the immune system to spot. The second, MSI, signals that the tumor's DNA is unstable. And here's the kicker. When you see high levels of both of those, it often means immunotherapy is going to work a lot better. Okay, so with all this evidence on the table, what's the final verdict on our suspect, CCT5? And, the really important question, what does all this mean for the fight against cancer? Well, the evidence is pretty overwhelming. It suggests CCT5 could be like a new Swiss Army knife for doctors. First, it could be a biomarker to help predict how a patient's disease is going to progress. Second, it could be a direct target for brand new drugs. And third, it could be an indicator that helps doctors decide who would benefit most from immunotherapy. That's huge. And the researchers behind this study? They completely agree. The case is strong. They concluded that CCT5 could be a powerful prognostic biomarker,
and a really promising target for immunotherapy. That basically confirms its status as a key person of interest in the world of cancer research.
And that leaves us with a really exciting question, doesn't it? This research has taken one of cancer's most reliable accomplices and turned it into a potential weakness, one we can actually exploit. So, could targeting this one single gene open up a whole new front in the war on cancer? For now, the case file remains open and the investigation continues.